So welcome everyone. It's good to see a lot of members participating in that first webinar of the series. I think we're gonna do a series of webinars in Polyphenols Research. So thank you very much for joining us. That's really part of the Groupe Polyphénols and as a follow-up on the ICP conferences that happened in Turku, Finland last year. So today we will have two presenters that will be introduced by Anne in a few minutes. And so we will have about 20 minutes of presentation by each of the presenters, and then you will have about 5-10 minutes maximum to ask any question you may have. So just as an info, so that webinar is recorded. That's because the recording will be available on the YouTube channel of the Groupe Polyphénols and on other social network of the Groupe Polyphénols. So if you have an issue with the recording, let us know. So now I will leave Anne presenting.
So good morning, good evening, good afternoon to all of you. I'm Anne Hagerman, Vice President of the Groupe Polyphenols this year. And it's our pleasure to welcome you to our first seminar webinar on polyphenols. We decided to do these to provide a little bit more inspiration to researchers in polyphenols, to introduce people to different areas of polyphenol research, and to try to generate that kind of enthusiasm that we get when we meet together every other year at the ICP. And for our first topic, we chose a topic that I think is really an up-and-coming topic in polyphenols research. It's an area that has received sporadic attention in the past, and it's really kind of poised right now to be a big area that could reveal a lot of really, really interesting information. And this is the idea of polyphenols in natural soil systems. So soil systems are a very, very complex system. Polyphenols enter that system, of course, from all the plants that grow in soils. And once those polyphenols enter the soil system, it's kind of a black box. We really don't know that much about what happens. It's been a very difficult nut to crack. It's a very, very complex system, lots of inputs and outputs and mysteries. But I think that with all of our new tools, the omics tools that allow us to use biology to tackle difficult problems, and the modern analytical tools that really allow us to delve into complex systems, I think we're really right on the edge of being able to make some great discoveries here. So to kind of inspire up-and-coming polyphenol chemists to turn their attention to soils, we've picked two scientists who can really give us some background and insights. And the first one that I'm really pleased to introduce is Inderjit. Inderjit has been a colleague for many, many years of mine. He is at the University of Delhi, where he received his education and has been on the faculty. And Inderjit has kind of balanced that fine edge of an ecologist with chemical interests who looks at both chemistry and ecology in these very complex systems. He's interested in chemicals in soils, and those are both human-made chemicals, and of course our natural chemicals like polyphenols, and how those chemicals interact with the soil ecosystem. He has a distinguished career with many publications, presentations, awards, fellowships to visit other universities. And so it's really our honor to be able to have him give the first seminar in our series. So Inderjit, we're happy to welcome you to talk about litter chemicals and mechanisms underlying vegetation changes. And if I can figure out how to stop sharing, or maybe Aude, you're taking over for me?
Yeah, Inderjit should be able to share his screen now.
Yeah.
Okay, yes. Inderjit, welcome.
Thank you very much, Anne, for this introduction. Since the time is 20 minutes, rather limited, so I will start right away. I will talk about plant chemicals.
Inderjit, could you make your screen bigger, please? Just click on slideshow. Sure. Thank you.
Is this okay now?
Yes, perfect.
Okay, I will talk about plant chemicals and their role in community organization. One of the core questions in ecology is how population are organized into higher, rather complex units or communities. One hypothesis, the Robotonov, first time gave a hypothesis that some plants are adapted to plant chemicals released by other plants into the environment. And this plays a crucial role, an important role in community organization. Plants produce variety of chemicals and then release into the environment. These chemicals influence, impact competitor, litter decomposition, enemies, herbivory, nutrient cycling, soil communities, and the different ecosystem factors and biogeographic evolutionary advantages plant is experiencing in the new ranges. Co-evolutionary relationship also impact the production, release, and ecological functioning of plant chemicals. Now, these chemicals, they play, play an important role in community organization at different spatial and temporal scale, from very small scale to large scale. But today, I am going to focus on medium scale by taking some examples from exotic plant invasions. Now, I will discuss the role of plant chemicals in community organization by discussing two situations. The first, origin does matter. Origin means origin of a species. The second, the origin doesn't seem to matter much. For first situation, I will take examples from exotic plant invasion. Some plants, when introduced to new ranges, novel ranges far from their home, they become abundant and impact plant biodiversity. They form monodominant community in the non-native ranges while they exist in species diverse community in their native range. Then I am going to discuss today the role of chemical explaining these two situations in the native home versus away. Now, when plants are introduced from the native range to non-native ranges, for example, here is an example of Ageratina. The older name of this species is Eupatorium, and home is Mexico. It is introduced to different part of the world. Here I am taking example of India and China, where this is a very aggressive invasive species. Now it comes here and introduce chemicals which are, I am not saying novel chemistry, but it is novel for invaded communities. Now, here in the home range, the plant exert weak chemical effects because the neighbors are adapted and microbes are co-evolved, but it exerts strong effects in the novel ranges because the neighboring plants are sensitive and soil microbial communities are naive. So this gives allelopathic advantage against resident species. This phenomena is known as novel weapons hypothesis, which is this name is picked from an event in the history in the 13th century when Mongol attacked Eastern Europe and won most of the parts in the Eastern Europe and also Germany. Then they analyzed, European army analyzed why they won. Because one of the reasons was they were carrying this novel weapon with the curved bow, and with this curved bow, they can fire arrow with greater speed and accuracy. So that's why this name novel weapons came. Very briefly, I explained when species move from native range to introduced range or novel ranges, what happens. The species experiencing negative feedback due to the presence of specialist enemy, which is not there, at least in the early stages of invasion. in the introduced range. So resources are allocated to the growth or the production of less costly chemicals. This is second thing which may happen is the novel weapons hypothesis, which I just explained. The third chemical related mechanism that may operate, plants experience negative feedback due to the presence of microbes. That's why natives uh, communities are species diverse, but this exotic plant experience positive feedback in the introduced range. And one example is accumulation of soil pathogens. So in most of these invasion hypotheses, the plant chemicals are involved. So here is the one of the good example of novel weapon. The exotic species Chromolina produce auditorin, which is in much lesser amount in their native range, Mexico, but very high amount introduced range China. Similarly, the other Chinese tallow, which produce higher amounts of quercetin, a flavonoid, in the non-native range US compared to its native range China, which also attract better, more mycorrhizal colonization. But on the other side, reverse may also happen. Here in the non-native range, production are more important in defense. Their concentration, their production is much less in non-native range compared to native range. This part is not clearly stated in novel weapon hypothesis. So we did a study with Agritina, the species I just talked. It is a strong smelling plant, and we show that the native species of native range and introduced range respond differently. The exotic range or introduced range native species, they experience more mortality compared to the native range. Then we try to isolate, identify the chemicals, mainly volatiles, terpenes and monoterpenes, by doing a common garden experiment, collecting seeds from native or introduced range. What we found was the two chemicals like 2-carine and alpha rendering. They produce in higher amount in the native range home, but low amount, but higher amount in the non-native ranges. But reverse is true for other four chemical, which is known for their role in defense like caryophyllene or beta-panin. So plant brings chemical. So there is a biogeographic variation in the production of chemicals. Some chemicals are produced in higher amounts, some chemicals are produced in lower amount. Then we also checked with another invasive species, what happens if a congeneric native species is present. Now in here, what we found that in Prosopis mesquite, Prosopis juliflora, which is exotic species, produce a chemical amino acid tryptophan. Now this compound is also produced by native species, but in much lesser amount, and native species is not able to accumulate this chemical in the soil. So effectively, the plant, exotic plant is building higher concentrations of this chemical tryptophan in the soil. So novel weapon hypothesis predict production of chemicals by exotic species in higher amount, and that provides the competitive advantage to the non-native species due to sensitive neighbors and co-evolved soil communities. We propose certain additions and refinement to this novel weapon hypothesis, that there may be biogeographic, native versus non-native variation in the production of chemical. The second, a plant may produce a chemical in the higher amount. It may happen that natives are also producing the same chemical, but the production is much higher so that the chemical can exert ecological impact on the neighboring species. This is coming again and again. Now, very briefly, I'll explain another very interesting Caribbean native, Chromalina. It has introduced to many parts of the world. Here in the Western Ghat, the chemicals released from its root, they uh, they seem to cultivate native soil pathogen. And we did a large number of experiments which show that we vary the concentration of chemicals released by the roots of this species. We did some activated carbon manipulation experiment, autoclaves, using autoclave soil and live soil. So what we found was that these chemicals manipulate, they cultivate the native soil pathogen, which then kills the seedling of native species. So we then gave this hypothesis accumulation of soil pathogen, which is mediated by chemicals. Novel chemicals are largely studied in context of novel weapons hypothesis or disruption of mutualistic association. But now, due to lack of time, I cannot discuss in detail, but they seem to play an important role in the mechanism behind most of the invasion hypothesis proposed till date. I hope I still have 5 to around 6-7 minutes. Now, here I discuss the second situation. Here, origin doesn't seem to matter much. What we have done is we have taken different tree species. Now, these tree species are taniferous tree species. They produce lots of condensed tannin. In fact, these species are introduced to Western Ghat due to their tannin producing abilities. In his seminal work, Paul Zinke in 1962, he provided evidence that tree species, they impact under canopy soil properties. And he has shown many different evidence to propose that this canopy effects on soil properties are important in community organization. We ask, what mediate the impact of these tree species on soil properties or soil processes? Then different tree species were taken. Here is the 6 different tree species. Origin is mixed. They may be exotic invasive, native species, native invasive, non-native, non-invasive species. And all these are, known to produce condensed tannins. Now here we quantify the tannins, tannin content, that the amount of litter they accumulate under their canopy. So what we found that, it is not dependent on the origin. For example, here, the maximum in Acacia melanoxylon, which is exotic, native of Australia, exotic, invasive. Then for this is native. This is again native, but invasive, then exotic. So the production of tannin or contribution or presence of tannin in litter is not origin here. Then what are the direct and indirect effects of tannins on soil nitrogen? So what we observe that litter condensed tannin of accumulated litter exert direct impact on soil organic nitrogen, but the indirect effects mediated by soil microbial activity are not significant, but total and direct effects are significant. On the other hand, the effects of condensed tannin on inorganic nitrogen, whether it's ammonium nitrogen or nitrate nitrogen, they are significant, mediated via soil microbial activity, and tannins, they do not seem to have direct effect on inorganic nitrogen. Then we were interested, to find out the direct and indirect effects on the under canopy diversity, total biomass, and total species richness. So what we found was the litter condensed tannin, they do have negative significant impact on total biomass and significant effect on soil nitrate and soil ammonium. Indirect effect on total biomass mediated via inorganic nitrogen. And in the previous slide, we have seen that this is mediated via soil microbial activity. But the total species richness, litter tannin doesn't seem to have an impact. It's not significant. The indirect effects are significant. Now here, when we analyze the under canopy biodiversity, impact on biodiversity, impact includes both positive and negative, the competitive exclusion and facilitation. So there are a number of species. Some species respond positively to the species. Some species are locally excluded. Some species are facilitated. So we believe that uh, total biomass is okay, but it would be more meaningful to examine the effect of a tree canopy on individual species. So for this, we found out certain species. Here I am showing the effect of how certain species respond to ammonia or nitrate. But today I am taking only example of ammonia. So these are different species which respond differently to soil ammonium, which is mediated indirectly by the litter tannin or tannin of accumulated litter. Here we can see all these species, they respond positively, but this Ageratina, Julex, Oplis menace, they respond negatively. So if this is the biomass. If we take total biomass, we may not have an effect. Some species biomass is increasing, some species biomass is decreasing, and uh, these species are exotic species. This is native of Europe, native of Mexico. So, I mean, more studies are needed to find out how the individual species respond to nitrate. We have done this study, but not yet analyzed the complete data and how they respond to organic nitrogen. What I believe that in future it is needed that we must do some validation experiment to uh, prove or disprove our hypothesis, what we observe in this field study. And the second, I don't know any study that has compared the impact of tannins on soil nitrogen in the native versus non-native range of a species. And we don't know whether there is a difference or no difference and how it behaves. So such study may generate some meaningful data. A second aspect, which we are very interested and should be done in the future, recently in 2017, the two papers were published in Science that shows that tree species, particularly in the temperate forest, they experience positive feedback. And they say that tree species experiencing positive feedback because they were associated with ectomycorrhiza. And most of those tree species produce tannin. Pinus was there and other species were there, the tannin producing species. So now, whether tannin mediate the positive feedback, uh, that we don't know, and how this mycorrhizae breaking down this complex nitrogen to simpler organic form to exert the positive feedback that should be studied in future. Thank you very much. I think 20 minutes are
Thank you, Inderjit.
over.
That was a really great overview for us as to how complex these soil systems are. I think in the interest of time, we'll go with no questions for this one. And when there are good questions in the chat, I'll just send them on to Inderjit and maybe you can have one-on-one interactions. But I do want to go ahead and introduce our next speaker, if I can share my props. So I'm really, really pleased to introduce our next speaker, whose topic really connects very well to Inderjit's last point there about the importance of microbial communities. Bridget McGivern is a doctoral student at Colorado State University, soon not to be a doctoral student, but to be a doctor, because her defense is coming up in just a few weeks here. It's really been my pleasure to be a mentor to Bridget as she's worked through her dissertation, and she's taken on some very challenging, complex problems. Her first problem was to look at condensed tannins in soil systems and to try to get some evidence about what happens to those condensed tannins when they enter soil systems. And she had quite a beautiful paper in Nature Communications last year about that. And she did receive the Ibrahim Award from the Groupe Polyphenols at the ICP last summer in recognition of that excellent work. And she has now gone on to try to extend her initial observations into other soil systems. And today she's going to talk to us about some permafrost and peatland gradients. This is really relevant because of the amount of carbon that these soil systems seem to control, obviously important to global warming. And also it's quite important because Bridget's data challenges the overriding paradigm about what happens to tannins in soils. And I think she's showing us that we can use these omics and high resolution analytical tools to really challenge old ideas and come up with new ones. So Bridget, we'd be delighted to hear about your recent work.
Awesome. Okay, let me share. Can you see my slides?
Yes, we can. They're great.
Okay, cool. Move this over here. Okay, cool. Well, thank you, Anne, for the introduction, and thank you to Aude and Denis and JP for organizing and inviting me to speak today. My name is Bridget McGivern, and I'm a PhD student at Colorado State University in the Wrighton Lab. And so our lab specializes in using high-resolution multi-omics tools to study microbiomes across environments, and particularly focuses on how microbial metabolism impacts ecosystem functions. And so my PhD, as you're going to hear, and like Anne kind of gave a great introduction to, is really aimed at studying how microbial polyphenol metabolism fits into carbon cycles, and particularly in soils. And this is a knowledge gap that has really important implications for understanding how soil will either store carbon or release it, and particularly in light of climate change. And so today I'm going to really jump into some new data that we have from the last chapter of my PhD, studying microbial polyphenol metabolism in a natural system. And so before we get much further, I just want to briefly touch on what the microbiome is. So a microbiome refers to the microorganisms, their genomes, and their surrounding environment that inhabit a given system. And so here, some of the players that you might hear about are bacteria, archaea, and viruses. Today, I'm really going to be focusing on bacteria and archaea. And my PhD is really focusing on the soil microbiome. So the soil microbiome provides many key services for the soil ecosystem. But the key thing to note for today is that the soil microbiome is responsible for many biogeochemical cycles. And so this illustrates some of these cycles. So for example, the nitrogen cycle, the sulfur cycle. But for understanding my talk today, really the key thing to know is this carbon cycle. So this is how the carbon cycle is traditionally presented, where plants take up CO2, use it to produce biomass, Then animals eat these plants, and when both the plants and animals die, they end up in the soil where they're decomposed and add to this stable pool of carbon. And in fact, soil stores more carbon than what's found in all of the plant material on Earth and all of the atmosphere combined. And this happens because the rate of carbon deposition by plants and animals exceeds the rate of decomposition. And so decomposition is really where the soil microbiome comes in. And this is really a simplification of all of the ways that microbes break down organic matter. And so here, this is a really simplified scheme of what we mean when we say decomposition. We're basically going from larger carbon, so polymers and monomers, that get broken down through central carbon metabolism of certain organisms, which can then feed other organisms by producing these smaller metabolic byproducts. And an important part of this is that both carbon dioxide and methane are often byproducts of this decomposition. And this is important because these are greenhouse gases. And while we often hear about CO2 because it's the most abundant greenhouse gas, methane is actually almost 30 times as potent as CO2 in warming. And so that's something I want you to keep in mind. And to make it even more complicated, some organisms can actually use CO2 and methane to fuel their own metabolism. And so the point of this really is that carbon metabolism is complicated, much more complicated than just this idea of decomposition. And we really need to understand how different carbon inputs will flow through this carbon cycle to ultimately either be stored in the soil or to be released as greenhouse gases. And so that brings me to our theme of polyphenols. And I'm really excited to talk to this group because I don't have to tell you that polyphenols are a really complex class of compounds. But something to keep in mind for this talk is that historically polyphenols have been lumped into this one group in soils. And so one of the things we're trying to do in this work is bring an awareness of the diversity and the complexity of polyphenols to this sort of more environmental field. And so with that in mind, I want to give you some background on the prevailing ideas about polyphenols in anoxic soils. Again, I sort of expect that you're all comfortable with the idea that polyphenols are important across ecosystems. And a comparison that I found to be really helpful during my PhD is to be thinking about human guts and soil systems, because in these systems, we know that polyphenols can play important modulatory roles. So for example, we know that the polyphenols we consume through our diet can give us health benefits. And in soils, and Inderjit gave a great introduction to this idea, we know that polyphenols can impact nitrogen cycling and also carbon cycling. And really at the heart of both of these roles is the microbiome. So in many cases, our gut microbiome has to break down the polyphenols in our diet to release bioactive metabolites that can go on to give us health benefits. And in soils, under oxic conditions, it's generally assumed that microbes can use polyphenols as a carbon source. But under anoxic conditions, it's assumed that microbes can't use polyphenols and that they might even be toxic to the communities. And so this interesting dichotomy on polyphenols and anoxic soils is really what I'm going to focus on today. And so now I just want to give an overview of the mechanism that's really behind this idea. So for decades, polyphenols have been thought to modulate carbon cycling through a mechanism called the enzyme Latch. And this has been supported by a number of really highly cited publications in high impact journals. The enzyme Latch is a biogeochemical cascade that explains how carbon is either stored or released from soils. In this cascade, the central assumption is that oxygen not only stimulates microbial growth, but is also required for the enzyme polyphenol oxidase. Polyphenol oxidase is important because in this theory, it's the only enzyme that can degrade polyphenols in soils. And this is important because polyphenols are thought to ultimately serve as a lock on soil carbon by either being directly toxic to microbial communities, shutting down their metabolism, or indirectly inhibiting them by limiting their ability to access substrates. However, this interplay between microbes and polyphenols in soils is really poorly defined and historically has been described by methods that aren't getting at the resolution at which microbial biochemistry is actually happening. And logically, it doesn't quite follow from what we know about microbes. And so, for example, the idea that polyphenol oxidase is the only enzyme that can process this diverse and abundant carbon pool doesn't really fit with what we know about the metabolic specialization that exists in the microbial world. And furthermore, as I mentioned, we know in our gut, which is an anoxic system, that our gut microbiota has to process the polyphenols we consume in our diet. And so isn't it possible that the same mechanisms that our gut microbes use could exist in anoxic soils? And so this question led us to conduct a study where we amended anoxic soil microcosms with a model polyphenol, so it was a condensed tannin, and tracked them over the course of 20 days with multiple high-resolution tools. And really the key thing to know about this study was that we showed as a proof of concept that anoxic polyphenol metabolism was possible by the soil microbiome. And we identified 10 candidate enzymes that we think were likely involved in breaking down this polyphenol, none of which were polyphenol oxidase. And I also want to highlight that we're by no means the only group that's been critical of the enzyme Latch. Nearly every part of this cascade has been critiqued. But for my PhD, the really interesting framing is that even in the studies that attack the enzyme Latch, this central relationship between microbes and polyphenol oxidase and polyphenols is taken for granted. And so building on our previous work, what I'm really now going to get into is how we're trying to examine this relationship at a field scale. And so the two questions I'm really going to cover today are, one, does the enzyme Latch hold in a natural peatland? And then beyond that, and building on our previous work, do we see evidence for polyphenol metabolism in this system? And so our field site for this work is Stordal and Meyer in northern Sweden. And so this is a permafrost peatland. Permafrosts are defined as soils that remain frozen for longer than 2 years. But with warming climates, these historically frozen systems are thawing. And so here in this image, you can see where this permafrost has thawed, leading to collapse, where this boardwalk used to sit on top of solid soil, but it's now submerged. Beyond the climate importance of this system, it's a really good model site for studying microbial polyphenol metabolism. And that's because there are 3 main habitats at Stordal and Meyer, which is created by this thawing permafrost. So there's the pulsa, which is the intact permafrost, the bog, which is the partially thawed part, and the fen, which is fully thawed. I'm depicting the system here in this image as it was in July 2016, which is when our samples come from. And so for this talk, the important thing is that this thaw gradient actually offers peat under a range of environmental conditions. And for this particularly, the important condition we care about is oxygen, where historical data suggests that the pulsa is oxic, and then as you move across this thaw gradient, it becomes more anoxic. Before I get into my data, the last thing I want to highlight are the methods that we use to study the microbiome. So first, we take all the DNA from the system and sequence it to get a metagenome, or a collection of all of the genomic information from a community. We can take this a step further and organize this data into metagenome assembled genomes, or MAGs, as I'll talk about them for the rest of this presentation, which really represent the genomes of organisms in this system. This information can provide us what we like to call a metabolic blueprint for these organisms and inform on the metabolisms that they could potentially encode. And while metagenomes tell us the potential metabolisms, we can use metatranscriptomes and metaproteomes to tell us what genes are actively being expressed by looking at all of the RNA transcripts or proteins respectively. And so in this study, we're really using metatranscriptomes, looking at the RNA. And we can ground all of this in the chemistry by looking at the metabolome, or all the compounds that we can extract and detect from the system. This informs us of all the possible metabolic substrates and products in the community. And so with that in mind, from this site, we obtained a suite of data from peat cores from each of our 3 sites, the pulsa, the bog, and the fen, at 3 different depths. And for this talk, I'm primarily going to focus on data from our metatranscriptomes. Also, we have pore water CO2 and methane measurements, and we also have assay data that mirrors the data that's traditionally been used to support enzyme Latch studies. So using this data, again, we had 2 questions. Does the enzyme Latch hold in this site? And beyond that, do we see any evidence for microbial polyphenol metabolism in Stordal and Meyer? So towards this first question, and a reminder of what the enzyme Latch cascade looks like, these are the relationships we would expect to see if this enzyme Latch theory holds, where oxygen and polyphenol oxidase are positively correlated, polyphenol oxidase and polyphenols are negatively correlated, and then polyphenols are negatively correlated with various soil biology parameters. And so what does our data really look like? I told you that we examined this using both sort of traditional methods, or the methods that have always supported the enzyme Latch, and more high-resolution omics approaches. So here I'm showing what those different data types show for the relationships between oxygen and polyphenol oxidase in this system. Here we're using depth as a proxy for oxygen, and plots are shown moving from deep to surface, or low oxygen to high oxygen from left to right. Interestingly, we support the enzyme Latch when we use metatranscriptome expression of polyphenol oxidase across depth, but not doing a classical polyphenol oxidase activity assay. Following the enzyme latch, we would expect to see a negative relationship between polyphenol oxidase and polyphenols if polyphenol oxidase was the only controller of polyphenol concentrations. And in fact, using both omics and traditional assays, we don't see a relationship here. Interestingly, we contradict the enzyme latch when looking at relationships between polyphenols and a proxy for microbial activity, as we see a significant and positive relationship between polyphenols and the number of transcriptionally active taxa in each site. We don't see a relationship between polyphenols and hydrolase enzymes, which is a really key assumption of the enzyme latch. We support the enzyme latch, showing negative relationships between polyphenols and carbohydrates across methods. And finally, and most excitingly, we contradict a key part of the enzyme latch and show that polyphenols in this site are actually significantly correlated with pore water CO2 measurements. And so when we step back and ask, what does this mean for the enzyme latch in a natural system? Our data shows that it's complicated, but on the whole, in our site, we see a positive relationship between polyphenols, microbial activity, and CO2 production, which pretty much refutes the enzyme latch. And so building upon this, our next question was, do we see evidence for polyphenol metabolism in this peatland? And so from this site, we have a database of 1,800 metagenome assembled genomes, or MAGs, that are detected across these sites. Here, I'm kind of giving us a window into each of these sites by representing each genome as a gray dot, where its size corresponds to its relative abundance in that compartment. Here, these dots are colored by their phylum, so that tells us who these microbes are. And we see basically a lot of red and a lot of blue, which corresponds to acidobacteria and actinobacteria, which are two phyla of really common soil bacterium. And so we wanted to know which of these organisms encode polyphenol metabolism genes. And actually, to answer this question, we needed to develop a tool for annotating polyphenol metabolism in microbial genomes. And so I want to briefly go over what annotation means. I told you that metagenomes give us metabolic potential, and a key step in inferring this metabolism is annotating. So in this process, we take a raw sequence of a gene and compare it to a database of known functions, genes with known functions. And then we look at the proteins that are most similar to our gene of interest and infer that because their sequences are similar, they likely have similar biochemical functions. After we've come up with a candidate annotation, we need to then put that newly annotated gene into a broader metabolism context. So in this case, for example, we can annotate this gene, see that it's phenyl monooxygenase, but what does that really mean? When we look at the broader context, it means that this organism can convert phenol into catechol. This is really challenging for polyphenol metabolism. I like to think of it as almost like a blind spot in our ability to annotate microbial genomes. And that's because, for example, a lot of the really cool biochemistry that's been done on polyphenol active enzymes hasn't been propagated into the databases we use to annotate our genomes. And so this means that we're basically never going to see these functions in our genomes. And even more so, if they are in our databases, it's often really challenging to infer the context, the metabolic context for these enzymes, because of either how the databases label them or because of the vast chemical diversity that exists in polyphenols. And so the average user who doesn't maybe know much about polyphenols will never be able to really put it into its important biochemical context. And so in light of this, and to address these knowledge gaps, we created CAMPER, which stands for Curated Annotations for Microbial Polyphenol Enzymes and Reactions. This is a computational tool that can annotate genes and genomes for polyphenol metabolism. To create this, I mined the literature for polyphenol enzymes and found 41 different genes that were biochemically characterized and carried out reactions across all of the major families of polyphenols. So there are all of these reactions shown here. And they're importantly not currently in databases. So in total in this tool, there are over 100 pathways, including the ones I'm showing here, which I created annotations for, and a further 300 genes that have been pulled from databases that I've reorganized to give polyphenol context. Furthermore, in this tool, there's information about oxygen requirements for these enzymes. And so we know from many of these enzymes discovered in gut systems, for example, that there are anaerobic polyphenol metabolism genes, here shown with orange arrows. And so basically, as a quick promotion for this tool will be publicly available. to both annotate your genes and give you context for these metabolisms. And so if you're interested and you have microbial genomes, this can be a tool you can use to infer that in your genomes. And so if you want to know more about this, you can definitely reach out and I'm happy to talk about it. So now that we have this tool in place to annotate polyphenol metabolism, we wanted to apply it to this data set of 1,800 genomes from this site to really ask who can potentially carry out polyphenol metabolism. Excitingly, we found that 83% of the genomes from this site encode genes for several polyphenol and phenolic active enzymes, which suggests that polyphenol and phenolic metabolism is really widely encoded in this site. And it paints a much more complicated picture of microbe-polyphenol interactions than just polyphenol oxidase being the sole controller of polyphenols. And so our next question was, since we see that it's widely encoded, do we see that there's active expression of these genes in the site? And really excitingly, we found that of the genomes that encode polyphenol metabolism, 53% of these are actively expressing these genes in the field. And so here the dots are scaled by their relative metatranscriptome recruitment, which is our proxy for basically how much gene expression they're doing. And you can see across these sites, particularly in the bog, that some of the more active members of this community are doing this. And again, we see a lot of red and blue, which corresponds to acidobacteria and actinobacteria. And we even see some archaea in the fen that are doing this. And really, this is important because it doesn't appear that polyphenol metabolism is like a specialized function carried out by a small fraction of the community, but rather it's widely encoded. And so I really want to highlight this. This is really cool. Not only do we see that polyphenol metabolism is widely encoded, but it's also widely expressed, and even in the so-called oxic and anoxic sites. And so there's a lot of really rich data here, but I want to highlight just one quick example that we've so far looked at in this metatranscriptome data as an example of the cool metabolisms happening in this site. And so what I want to highlight is caffeic acid reduction. And so caffeic acid is a phenolic acid, and there's a reduction system that's encoded by the CAR operon. And so this is a really interesting metabolism because microbes that do this couple the oxidation of hydrogen. Basically, they use hydrogen as an electron donor and caffeic acid as an electron acceptor. So this is really a truly anaerobic metabolism. Here, I'm showing you the expression of caffeic acid reduction across this site. So the bar plots here are summed by the transcription of the CAR operon across these sites and are colored by the phyla of the organisms that are expressing these genes. And the numbers at the tips of these bars correspond to the number of different genera that are expressing this system in each compartment. And so we see this metabolism expressed across habitats and depth. And most interestingly, we see that it's expressed by 86 different genera across 8 different phyla. And to give you some context on why this is so impressive, this metabolism has been around since the 1980s. And in the decades since, it's been biochemically characterized in just a single model species. And so here we're showing that it's likely much more broadly encoded in that single model species, and in fact is encoded across 8 different phyla. And so I want to close with like 4 reasons why I think this is an interesting metabolism. So for one, like I said, this was previously limited to just a model bacterium, but here we're showing it's likely conducted by several other types of organisms. 2, it's a true anaerobic metabolism. So this model where polyphenol oxidase is the only thing that happens and polyphenol metabolism shuts down under anoxic conditions just doesn't hold. It's also an interesting example because in this case, the polyphenol is actually serving as an electron acceptor. And so polyphenol metabolism is more complex than just a polyphenol being broken down and serving as a carbon source. Here's an example where it's actually supporting respiration. And finally, and maybe most sort of interesting, is that caffeic acid is often cited, including explicitly in some of these enzyme Latch papers, as being microbially toxic. And here we're showing it actually is not toxic and might be supporting growth for these organisms. And so to answer this question, not only do we show that the enzyme Latch doesn't hold in this system, but we've shown that polyphenol metabolism is active by diverse organisms and diverse members of the community, regardless of redox. And so to close, I just want to return to this idea of decomposition in the soil microbiome. And so we have a lot of data to dig into still, including linking this metabolism back to CO2 and methane production in this site. But I hope this has given a good description of how we're really working to put polyphenol metabolism and basically take it beyond the enzyme latch and say this is where it belongs within microbial carbon cycles. And so there's been a lot of really awesome people that have been involved in this work, but I want to specifically acknowledge my PhD advisor, Kelly Wrighton, and also Professor Anne Hagerman, who's been a mentor and advisor to me on all things polyphenols. And so with that, I'm happy to take any questions. Yeah.
Thank you, Bridget. I think that really ties it together from the chemistry side from you and the ecology side from Inderjit. And actually, we do have one question, which is the same as my question. Is Camper published yet?
So we have it it's available right now on GitHub, and we're still in beta testing with some people outside of basically me to make sure that other people can use it and interpret the outputs. But the plan will be by probably June that it'll be publicly available.
Okay. I think it is so cool the way that you can use that essentially biological information that comes from the omics to figure out chemistry. You know, from my old background, chemistry was going to explain biology, but I think you're kind of turning things upside down there. So I think that closes our formal presentations, and I believe that Denny is now going to take over and give us some closing comments.
Okay, so thanks a lot for giving me the opportunity to talk to you. I am sorry I was a little bit late at the beginning of the seminar, of the webinar. I had connection problems, so this is probably the only thing that didn't work completely well today. So really, it was an important day for the life of the group Polyphenol, since this was actually our first webinar in polyphenol research, the first of we hope a long series. But of course, it's not it. So first of all the presentations will be made available to you, even for the colleagues that, for example, who could not attend the webinar today. And the presentations will be made available on the Groupe Polyphénols website, on the YouTube channel of the Groupe Polyphénols, and also on the Instagram and Twitter accounts of the Groupe Polyphénols. Also, a second important thing is that you will receive soon a survey where you will have the opportunity to give us some feedback about the webinar. It was the first one, so certainly a lot of things to improve. So, but also things that work very well. So please don't hesitate, comment, and this will, of course, help us to improve. As I told you, this is the first seminar of hopefully a long series, and we plan actually to give 2 additional webinars in 2022, probably in September and November. And the possible topics that will still need to be refined depending on the acceptance of the people who will present. The first one could be the phytochemical characterization. The first topic could be around the phytochemical characterization of complex mixtures. And the second one in November or December, could be the polyphenols and food and beverage quality. Okay, so before I close the session, I wanted, of course, to thank you all for having attended this seminar. I think that we are close to 50 attendees, so it was really a big success. I hope you enjoyed it. Of course, I would like to thank our 2 speakers who kindly accepted to give a presentation today, Inderjit, of course, and Bridget. And for Inderjit, it was quite late today, and Bridget quite early. So thanks a lot for having given your presentations. Thanks a lot to you, Anne, for having animated this session on the scientific point of view. And finally, of course, the preparation, the setup of these seminars was quite a substantial work. And actually, we would not be there at all, and probably nothing would have happened without the help of Aude, who organized all the WebEx session, and the help also of Professor Juha-Pekka Salminen, who is our communication manager and will take care, for example, of the recording of the webinars, and will put that, of course, on all the sites I was mentioning before. So yes, thanks to everybody, and maybe now, unless there are other burning questions, we can close the session. Do you want to say a word, Aude?
No, I just sent in the chat box, thanks to everybody. And thank you, Inderjit. Thank you, Bridget, for your great presentation. And if you have any questions, feel free to send directly an email to the presenters if you want to learn more, or to any of us.
Okay, so I think that we can close the session now, and you will soon receive the information concerning the links to the videos, and of course, information about the next webinars. Bye-bye.